All right, hello, biology people. Here to finish off our last little bit. We've covered most of the material on diffusion, on osmosis. This next bit will conclude our discussion on chapter 6. So we get now into the idea of active transport. So we've looked at the idea of concentration gradients going from high concentration to low, either through the direct or directly through the membrane, simple diffusion, or through our IMP, our integral membrane protein. So again, you should be knowledgeable on the phospholipid bilayer, the hydrophilic, hydrophobic, hydrophilic areas, our IMPs, concentration gradients, tonicity, hypo, iso, hyper, ATP, passive transport, all that is kind of what we've covered before. So now we focus on this bottom idea that we call active transport. So the movement of particles from a low concentration gradient to a high concentration. So if you remember the good old days when you used to kind of play on the slides, if you were very small, you don't even have to climb up. The parents just set you up top, and all you do then is slide. You kind of go from a high to a low concentration. So this is sort of representative of sort of going from a high to a low concentration area. So the left picture here kind of represents sort of osmosis, diffusion, passive. You don't really have to put a lot of energy once you're here. You just kind of slide down, go for the right. Some of you, this was not exciting enough. So you start at the bottom and you try to climb up against the sort of against gravity, against the normal sort of flow. So this requires more effort. It's not necessarily something passive. You really have to work hard. You're sliding and moving up and sliding down and putting a lot of effort to go back to the top. So the picture on the right-hand side then representative of our active transport situation. So sort of diffusion on the left, active transport on the right. So again, the official definition here being the movement of molecules from a low concentration area to a high concentration. So again, the opposite of diffusion, low to high. We're moving against the gradient. So if we were to open this channel, the high concentration wants to diffuse in. So we want to go from right to left, but no. We don't let that happen. We go from left to right. So if you look at this picture here, here's our phospholipid bilayer. This sort of purple sort of oval, which they call a proton pump, is an IMP, right? It's an integral membrane protein, but it functions differently than these facilitated diffusion proteins. So it's not allowing diffusion to happen. It's actually pumping with the help of energy, ATP. We're pumping this energy, we're pumping these hydrogens from right, I should say from left to right, from inside of the cell, outside of the cell. So in order for this to happen, it will require this energy form ATP. So if we look at the idea, the goal, the overall purpose of active transport, right? It is to not allow equilibrium. If we go back a little bit and we look at the goal of diffusion, diffusion's goal was to achieve equilibrium. So this is a very different thing. We don't want balance, right? I have a picture here of a wasp, right? There's wasps outside in the world, right? And there's none inside your home. But if we were to have balance, if we were to allow equilibrium, we would have the same number of wasps outside as inside. If you've ever been driving and a wasp comes into the vehicle, you're flapping your arms, you're trying to shoo this bee or this wasp out, right? That requires energy, and that then is trying to represent, we don't want it to be here, we want to force it out where there's a bunch, right? If there's flies in your home, the same situation. I have a picture of a bat. I don't know if you've ever had a bat flying around your house. It's quite interesting. But You do everything you can to try to get it sort of out, right? Go out. We don't want you to reach equilibrium inside the home. We want out. If we can go back to chapter 2, right? Those of you A students, right? You students that are piecing everything together, that are understanding. So what is going on here, and why are we pumping out hydrogens from left to right? Why do we just not allow equilibrium? Allow these hydrogen cations to diffuse in? Why do we not allow that to happen? I hope you remember the concept of acidity, of pH, right? pH, the potential of hydrogen cations. If we allow all of these hydrogen cations to come into the cell, we start to increase the acidic level. The cell starts to become more acidic. Our pH value gets smaller, right? Remember, as we go from 7, 6, 5, 4, 3, 2, that becomes a greater, it's an inverse logarithmic kind of situation. We become more acidic, and we push that that balance. We go farther from neutral, and that starts to have a negative impact on our, again, our proteins can start to be denatured, and they don't function very well. So we don't want the acidity to come in. We're trying our best to throw that acidity out, right? These hydrogen cations, kind of like when you throw the garbage, right? If you analyze your water bill, maybe your mom and dad pay the water bill, but there's a fee there that you pay so they can come pick up your garbage and take the garbage away. So you pay money so they can haul off your garbage, right? We're not talking about money here, but we're talking about energy. So the cell will expend energy so the garbage can be hauled out and pushed out. Where does it go from there? I don't know. We don't care as long as it's not in here. So the idea of active transport, a very important sort of mechanism, opposite of diffusion, to help sort of keep a gradient, right? Diffusion wanting to diminish the gradient, active transport wants to keep the gradient going. So this is a good test question here, right? So when would active transport stop? When we reach equilibrium? No, we don't want to reach equilibrium, right? Active transport will stop when we run out of ATP. When would osmosis, when would diffusion stop? When we run out of ATP? No, because we don't use ATP, right? So we would stop during equilibrium. When we are in diffusion, we would stop when we run out of ATP, when we're dealing with active transport. So pretty good concepts that I would know if I were you. You're probably going to see those on the next exam. So again, differentiate those concepts, active transport and diffusion. So the last sort of process, I don't know what you want to call it, phenomenon, process by which we can move products out of a cell or into the cell, is the idea of endocytosis, right? Endocytosis. In 1306, we use the term endocytosis. In anatomy 1, anatomy 2, you'll use the term phagocytosis. It's the same concept, just different vocabulary. So when we deal with endocytosis, the key aspect here is large particles. So we're trying to move something into the cell and across the membrane that's too large to fit through the IMP. So, or if it's not too large, it has the chemical characteristics that don't allow it to pass through an IMP. So it's like trying to move something, a big sofa, in through a little door. It's not going to work, right? And we don't have to take off or break the wall, right? Because our membrane is fluid, right? Our membrane has that plasma aspect to it. So the membrane can be shifted and modified to allow the movement of large particles in, right? In this process we call endocytosis. It reminds me of that big hug, right? So see if this makes sense. So they're distinguishing here that first layer of phospholipids, kind of a yellowish color, and then a darker orange color, sort of this other layer. So hydrophilic, hydrophobic, hydrophobic, hydrophilic. Now we're pushing these large particles and the actual membrane starts to kind of fold inwards a little bit. And if you notice, we start to kind of make a little compartment here. And then there's a reshifting, a reconfiguration of our actual plasma membrane to then trap these little components inside. So here's our yellow, 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 and then orange, 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 orange. So we have modified the original membrane. We took kind of like a little miniature membrane and we put it inside of the original membrane. I'm going to talk about these little membrane enclosed aspects in the next chapter again. But for now, just understand the movement of these particles by this process, this sort of enfolding and restructuring a membrane in 3 dimensions all the way around, we would call endocytosis or phagocytosis. So same concept, different picture. We have some large cucumber looking thing here that starts to push on this little blue line representing our plasma membrane. And plasma membrane starts to enfold. And then we kind of make that little reconfiguration there. And we seal off with a membrane inside of the original membrane, right? Bringing the large particle in, right? Endocytosis. So endocytosis, bringing large particles in. And the opposite process we call then exocytosis, like an exit. You're leaving, you go out through the exit. So we have the sort of little internal membrane that reconnects, configures with the original membrane and releases the contents out without leaking out any other important material from the inside. So endocytosis or phagocytosis in, exocytosis out. So again, exocytosis being the reverse process of endocytosis. So right now we see that yellowish layer of phospholipids is towards the inside. And when we reestablish, it goes back where that yellow layer of phospholipids is now facing the outside of that cell. So it's a neat trick that these cells can do to move particles across, in or out, depending on the situation. So a good summary slide here. What are 3 ways that a cell can move particles across its membrane, right? 1, diffusion. And we have a couple of sort of ideas: diffusion, simple diffusion, or facilitated diffusion. With diffusion, we have our tonicity, hypo, iso, or hyper. Trying to achieve equilibrium, passive, no ATP. We see the opposite process here, looking at sort of an active process moving against the gradient from a low concentration to high concentration, not trying to establish equilibrium, trying to prevent equilibrium. And then the special case movement in or movement out, exo out, endo in, of large particles or particles that don't or not able to pass through these IMPs. So again, potential essay questions that I can take from here, lots of multiple choice questions that I can derive from these as well. So this last slide that I want to kind of finish up chapter 6 with tries to integrate and also foreshadow a little bit, but integrate a lot of the concepts that we've been covering. And specifically, we're looking now at these proteins. So we cover proteins in chapter 3, and now we integrate them into chapter 6. We know that they can serve as transporters, this facilitated diffusion or active transport sort of proteins. They can allow materials to pass from one side of the membrane to the other. Within the actual membrane, they can serve as enzymes. So they can basically catalyze reactions from their position within the membrane. These IMPs can also serve as sort of key lock mechanisms. We know that insulin is able then to open up some membranes or open up some of these IMPs for the movement of glucose. Vitamin D can open up for the movement of, let's say vitamin D for calcium movement, yeah? So these IMPs also serve as a connection spot for oligosaccharides, for cell recognition, cell marking. We're going to look in the next chapters how IMPs can also serve as connections for one cell to connect to another cell. So these cell adhesion proteins, and we can also see connections within the cell. So cytoskeletal components from within the membrane. So with that, that will conclude our discussion on our chapter 6. So I'll get this posted, and if hopefully you can finish up the homework on Pearson as well. Right? So with that, y'all have a great evening, and I'll see y'all next time.